All right, welcome to The Explainer. Today, we're going to put on our detective hats to solve a fundamental genetic mystery. It's a case that really gets to the heart of how traits get passed down from one generation to the next. We all learn the basics in school, right? Mendel's laws. They tell us that genes for different things, like, say, eye color and hair color, are inherited totally on their own. But, genetics, just like any good detective story, is absolutely full of exceptions. So what happens when genes don't play by the rules? What if they kind of stick together? Well, that's the very case we're cracking today. Okay, so let's just lay out the facts here. On the one hand, you've got independent genes. The best way to think about it is like reaching into a bag and pulling out two different colored marbles. Getting a blue one doesn't change your odds of getting a red one at all. But our mystery is all about linked genes. These are more like two beads that are physically stuck together on the same string. If you pull one out, you better believe the other one is coming right along with it. They're connected on a chromosome. All right, let's dive right in. Our first case file, it involves some very specific rabbits and their babies. And this is where we're going to find our first big clues. So the scene is set with a classic experiment, a dihybrid cross. Scientists are looking at two traits at the same time, whether a rabbit's coat is spotted or not, and whether its hair is short or long. Now, they were probably expecting to see Mendel's laws play out perfectly. But they found something else, something much more interesting. And here it is. The evidence from this cross gives us our first real hard clues. Just look at the numbers. You see those two big groups, the parental types? They look exactly like the original parent rabbits. But then you've got these two tiny little groups with new combinations. That huge difference in numbers, that's our smoking gun. Now here's the thing. If these genes for coat pattern and hair length were truly independent, we'd expect all four of these combinations to show up in pretty much equal numbers. But that's so clearly not what we're seeing. The fact that most of the offspring look like the parents strongly suggests they're inherited together. So, are the genes linked? All the evidence is screaming yes. In any good investigation, you follow the evidence. And here, the most important clues, the ones that are going to crack this thing wide open, are actually hidden in those two groups with the smallest numbers. They're the exception to the rule, and they're exactly what we need to figure out just how sticky these genes are. So these super rare offspring, we call them recombinants. And they can only exist because of a process called crossing over, which is where chromosomes literally swap little pieces of themselves. Think back to our beads on a string. Crossing over is like the string breaking and getting retied in a new way, creating a brand new combination. And how often that break happens tells us exactly how far apart the genes are. And what's so cool is that we can turn these clues into a real number. It's just a bit of simple math. Let me walk you through it. First, you count up those recombinants. 10 plus 7 is 17. Then you get the total number of offspring, which is 158. You divide the small number by the big one, multiply by 100, and that's it. And boom, 10.8 map units. Now, this isn't a physical distance. You can't measure it with a ruler. It's a genetic distance. It basically just represents how often these two genes get separated by that crossing over event. The bigger the number, the further apart they are, and the more likely they are to be separated. For our rabbits, case closed. Okay, so we've cracked the case for rabbits. Now let's take these detective skills and apply them to something with much higher stakes: human genetics. The thing is, we can't just set up experiments with people. We have to work with the evidence that families already give us. So when it comes to humans, our number one tool is the pedigree. It's really just a fancy family tree that lets us track a specific trait, or more importantly, a genetic disorder. You've got squares for males, circles for females, and the shading tells you who has the trait. By digging into these patterns, we can find linked genes in us too. And check this out. Here's a real-world case file. Nail patella syndrome. It's a rare disorder that affects, well, your nails, kneecaps, and elbows. And by studying family pedigrees, scientists started noticing something really interesting. It was almost always inherited along with a person's ABO blood type. That discovery proved that the gene for this syndrome is physically on the same chromosome, chromosome 9, as the gene for blood type. And this is the whole point, really. Just like Dr. El-Zaweli says here, by looking at these family trees,
Scientists can do the exact same math we did for the rabbits. They can calculate how often these traits recombine and use that to build maps of our own genes on our chromosomes. This is absolutely critical for medicine.
So we've seen how this all works for rabbits and for humans. But it kind of makes you wonder, doesn't it? This whole incredible process, it raises a much bigger question. And the answer is honestly pretty surprising. I mean, where did all these amazing detective tools even come from? Who were the original code breakers who figured out recombination and gene mapping, and even the basic idea that DNA is what carries our genetic information? The answer is probably not what you're thinking. That's right. Believe it or not, our deepest understanding of genetics didn't come from studying people or rabbits or fruit flies. It came from the microscopic, hidden worlds of bacteria and the viruses that infect them. These incredibly simple little things were the Rosetta Stone that let us crack the entire genetic code. So why bacteria? I mean, of all things. Well, it turns out they're the perfect little informants. They reproduce like crazy, they're super simple, they're small, and it's really easy to see when their genetics change. This meant early scientists could run experiments that covered generations and generations in just one afternoon. That's how they uncovered the fundamental rules of life. All right, so let's get into the nitty-gritty of the toolkit these original codebreakers gave us. It turns out bacteria and viruses are just constantly swapping genes back and forth. And by figuring out how they do it, we learned how genetics actually works at the molecular level. Now, viruses have two main ways of operating. The lytic cycle, that's basically a smash and grab. The virus gets in, makes a ton of copies of itself, and then bursts the cell open, destroying it. But the lysogenic cycle is way more sneaky. The virus just slips its own DNA right into the host's chromosome. It hides out, getting copied every time the cell divides. And that ability to merge DNA is key. So how do bacteria get new genes? Well, the first method is called transformation. A bacterium can literally just slurp up a piece of DNA that's floating around in its environment, maybe from a cell that died. It's kind of like finding a random blueprint on the street and deciding to add that feature to your house. A little weird, but it works. The second method, conjugation, is way more direct. One bacterium actually builds a tiny little bridge to another one and passes a copy of its DNA straight across. It's pretty much the bacterial version of plugging two computers together with a USB cable to share a file. And finally, we have transduction. And this is where our viruses come back into the picture, acting like an accidental genetic delivery service. A virus might be assembling itself inside a bacterium and accidentally grab a piece of the bacterium's DNA. Then when that virus goes to infect a new cell, it injects the old host's DNA right into the new one. So think about this journey we've been on. We started with some sticky genes in rabbits, moved to solving human medical mysteries, and we ended up in the secret gene-swapping world of bacteria and viruses. The principles discovered in these tiny organisms gave us the very tools we needed to map the entire human genome. So now we have the map. The only question left is, what mysteries are we going to solve next?